Expecting a baby comes with a mix of excitement and a very specific kind of anxiety. You're constantly wondering about the hidden health of the life growing inside. To understand what's actually happening in there, we have to look close. Think of human biology like a factory. To build a healthy baby, that factory requires a very specific set of instruction manuals, exactly 46 of them, organized neatly into 23 pairs. Biologists call these manuals chromosomes. Every single microscopic cell in the developing embryo acts as its own factory floor. These cells constantly read from those 23 paired manuals to know exactly how to form the heart, the lungs, and the brain. Human development is an operation of absolute precision. If a cell has 46 chromosomes, the instructions are clear and development proceeds normally. That exact number is the biological baseline for a healthy start. But sometimes a sorting error happens when cells divide. Biologists call this non-disjunction. Instead of the chromosome pairs cleanly separating, one pair sticks together and both copies get pulled into the exact same cell. This creates a condition known as aneuploidy. The resulting embryo inherits an abnormal number of chromosomes. Instead of the standard 46, it ends up with 47. If that extra chromosome lands on the 21st pair, the result is trisomy 21, commonly known as Down syndrome. This extra set of instructions leads to specific physical differences, like a flatter facial profile, a single palmar crease on the hands, and an increased risk of congenital heart defects. If the extra chromosome lands on the 18th pair, it causes Edwards syndrome. The physical impact here is more severe, often resulting in tightly clenched fists, rounded rocker bottom feet, and life-threatening organ defects. Because a single microscopic packing error severely disrupts a child's medical future, doctors needed a way to look at the baby's genetic code long before birth, while it is still completely hidden inside the womb. Around the 10th week of pregnancy, doctors can offer the first major look at the baby's genetic health. It's called non-invasive prenatal testing, or NIPT. As the pregnancy progresses, tiny fragments of the baby's DNA break off from the placenta. This cell-free fetal DNA drifts directly into the mother's flowing bloodstream. By taking a simple blood draw from the mother, labs can isolate those tiny floating fetal fragments. Modern sequencing machines then map and count millions of these DNA pieces one by one. If the computer algorithms detect an unusually high concentration of DNA mapping back to chromosome 21, the digital tally spikes, signaling a high risk for Down syndrome. NIPT gave doctors and parents the ability to gain deep genetic insights from a routine blood draw, meaning there is zero physical risk of miscarriage to the developing baby. But NIPT comes with a critical limitation. It is strictly a screening test, not a definitive diagnostic test. Think of it like a weather radar. The radar looks at clouds and predicts a high probability of a storm in your area, but it cannot guarantee with absolute certainty that rain is currently hitting your specific roof. False positives do happen. Sometimes biological quirks isolated solely in the placenta will release abnormal DNA fragments directly into the mother's blood, triggering an alarm even when the baby itself is perfectly healthy. A high-risk NIPT result isn't a final answer. Instead, it prompts a pivot in the medical journey, shifting the focus away from probability screening and toward the pursuit of complete diagnostic certainty. To get that absolute certainty, doctors have to bypass the floating blood fragments and look directly at actual intact fetal cells. They obtain these cells through an invasive procedure like amniocentesis. A medical professional carefully inserts a fine needle into the abdomen to extract a small sample of the amniotic fluid surrounding the baby. Throughout the entire process, doctors rely on an active ultrasound feed. This ensures the needle stays entirely safe and completely clear of the developing fetus. Once that fluid is collected, the lab has the direct physical material it needs. Scientists can now read the baby's genetic blueprint without any guesswork. The traditional gold standard method for analyzing those cells is called karyotyping. Scientists culture the cells in a lab and then physically inspect the chromosomes under a microscope. This karyotype map organizes and numbers chromosomes into pairs from 1 to 22. An extra chromosome, like in trisomy 21, is immediately obvious. However, this map has low resolution. Because scientists look at overall shape, microscopic errors hidden inside these blurry bands remain invisible. For hidden errors, modern labs use chromosomal microarray analysis, or CMA. It acts like a high-powered digital zoom lens, revealing the genome as a barcode-like sequence. It scans the DNA to spot microdeletions, minuscule missing chunks of genetic code that karyotyping would easily miss. NIPT serves as the safe, early warning radar, while karyotyping and CMA provide the definitive truth. By achieving this level of diagnostic clarity, parents and doctors can transform a moment of uncertainty into a precise medical plan, ensuring they are fully prepared to provide the best possible care when the baby arrives.